When DNA gets copied, who gets to keep the original strand? This is the question answered by Meselson and Stahl in their classic 1958 experiments when they show that DNA replication is semi-conservative. This has nothing to do with DNA's political leanings. Instead, it has to do with the fact that when DNA gets copied, one strand is used as a template for the other strand, but after they split, then after that first split, they're not coming back together. So when the DNA strands separate, one strand is being used as a template for making the other strand, then those separate. One strand is being used to make it as a template for making the other strand, and then those separate, and etc., etc., etc. This is in contrast to like a conservative model, whereby the strands of DNA would separate, get copied, then those would the new copies would leave, and those originals would stick back together. Or a distributive model. I mean dispersive instead of distributive. I don't know why I had it in my head as distributive, but it's dispersive, and so sorry about that, and I do that a couple times. Where like parts of them would be used, like you have parts of a new strand and parts of an old strand and parts of a new strand and parts of an old strand. And so this is, how did they figure this out? You can imagine that it's kind of like if you were to have a flyer, and it was like a 2-page flyer, and one flyer has the encrypted message that'll tell you how to like write the second flyer, and the second flyer has the interpreted message, or like the encrypted message for the first flyer. I'm going through this technical like confusion because DNA has this like reverse complementary thing where the strands are running in opposite directions. We call these anti-parallel, and the strands are complementary, so they're not identical, but instead it's more like if you have a G on one strand, there's a C on the other strand. If you have an A on one strand, there's a T on the other strand. So the strands aren't identical, but if you have one strand, you can use it as a template for making the other. So we have this flyer, and we highlight these original copies. And if you've ever gone to Xerox something that's been highlighted, you'll realize that the highlighting doesn't copy, which can be good or bad depending on whether you wanted that highlighting to be copied or if those were just notes for yourself. But anyway, so you have these original copies, and you make copies of them, and then those copies are not going to be highlighted, but your originals are still going to be highlighted. So now what's going to happen? Are those originals, do you end up with the originals coming back together? Do you keep the originals together and then each of these new ones is being used as a template? Or do you then keep one strand of the old and one strand of the new? So you have one highlighted, one non-highlighted, and then those will get used to make copies. So then those will get separated, and then each of those will be used to make a copy. Those will get separated, each of those will be used to make a copy. End result, your originals get separated in that first place, and then every cycle they're getting separated again, and so they're going to get like a new strand each time, but your original strands are still going to stay intact. So if you imagine, if you had a model that was semi-conservative, this is what you would see. If you had a model where it was conservative, those originals would stay together until the very end. So you would keep separating them to make copies, but then they would be coming back together. Another option is that it would be like distributed, and this would result in there being like pieces of the new and pieces of the old, and so you would end up with you'd have highlighting in some portions on some text, and some portions on the other's text, and some portions on one text, and some portions on the other text. And so this might seem like totally like, how would that happen with DNA? But it was actually promoted by some prominent scientists because it seemed like it would make sense, because you have these two strands of DNA. In order to actually access and template them, you would have to like pull them apart, and so they thought if you did this in like little pieces, it would be easier. And so you can kind of imagine like cutting DNA and then like making these little pieces and then like cutting together and stuff. But instead, this process we show, like now we know that there are enzymes, there are like topoisomerases that actually help like make little nicks and then like unwind things so that there's space that the polymerase can do its thing, but the strands are going to stay together because we don't want to break up our DNA, because then that can cause all sorts of problems. Instead of highlighting pieces of paper, Meselson and Stahl were doing this by using heavy isotope labeling. So they were growing bacteria in heavy nitrogen, which we'll talk about. And so this version of nitrogen is heavier, so the DNA that gets made, because DNA has nitrogen in it, it's going to incorporate this heavy nitrogen, and then these DNA strands are going to be heavier. When this is going to allow them to separate heavy and light containing DNA using a centrifugation method with like a cesium chloride gradient. More about this stuff in a minute. But in this way, they're able to separate the heavy from the heavy light from the light. And what they see is that in the beginning, they have all heavy because they've grown it in this heavy medium. Then they in the next cycle, they have heavy light, where one strand is heavy and one strand is light. As you go on and on, you're going to have more and more of the pure light, but you're still going to have a little bit of that heavy light because the original copies are never coming together, and so you're never going to get heavy again. But that original copy is going to stay there because it continues to be used as a template. It's just kind of getting like diluted out.
So this is the paper that we're going to be discussing. It was published in PNAS in 1958. It's open access, which means that it's free for anybody to read. And it's a fairly short, concise, nicely described article. And we'll be going through some of the figures and some of the experiments that they did. But first, let's start with a little background about the techniques that they're going to be using. So their experiment is going to be relying on this idea of nuclear isotopes. So atoms, like those individual nitrogens, carbons, hydrogens, oxygens making up the DNA, are made up of smaller parts called subatomic particles. So these include protons, neutrons, and electrons. The protons are positively charged and they hang out with the neutral neutrons in the nucleus. So this is like this dense central core of the atom. And then there's this cloud of negatively charged electrons that whiz around it. We tend to pay the most attention to these electrons because atoms can share pairs of electrons and kind of merge parts of their electron clouds in order to form these like strong bonds, covalent bonds that are making up molecules. And if those electrons shift around, they can get like partially charged regions that then make the atoms want to hang out together. So we can get like non-covalent interactions. So we pay a lot of attention to these electrons, but it's really the number of protons that's actually going to define the element. So nitrogen is always going to have 7 protons, carbon's always going to have 6 protons, oxygen's always going to have 8, hydrogen's always going to have 1. So if you have an atom and it has 7 protons, it's a nitrogen. So if you have an atom and it has 7 protons, it's a nitrogen. But the number of neutrons and the number of electrons can vary. If you change the number of electrons, you're going to change the charge because the electrons are negatively charged. If you change the number of neutrons, you're not going to change the charge because these neutrons are neutral. But you're going to change the mass because these neutrons have mass. So electrons are so tiny, we typically don't even think of them as having a mass, but the neutrons are like the same mass as the protons. And so if we add a neutron, we're adding weight. And when we're adding weight, we're also we potentially destabilizing the nucleus. So those neutrons, if you thought we ignored the protons, we really ignore these neutrons most of the time because they're neutral. And what they're doing is they're really helping glue together these positively charged protons. If you imagine you have a lot of positive charge, same charges repel each other. And so the neutrons are really there helping provide this glue that's holding together this nucleus. It's a fine balance. So if you have an imbalance of the amount of protons and neutrons, you can get a unstable nucleus. And if this nucleus is unstable, then it can stabilize itself by giving off radiation. So it can undergo radioactive decay, where basically it kind of rearranges itself, kicks out some particles, and has a more stable nucleus. But there are also cases where you can add neutrons and then it doesn't affect the stability, or at least not enough to actually matter. And so then in these cases, your atom is going to be heavy. So it's going to be heavier because you have more neutrons, but it is stable and it's not radioactive. So you can have different what we call nuclear isotopes of the same atom, where you have different numbers of neutrons. And these nuclear isotopes can be stable, so they can be heavy but not radioactive, or they can be unstable and be radioactive. With nitrogen, so like the nitrogen we normally think of, and the light nitrogen that's going to be referred to in this paper is going to be nitrogen 14. The 14 comes because it has 7 protons and 7 neutrons. It also has 7 electrons in its neutral form, but we're not going to think about the electrons because when we're talking about the mass, we only care about the protons and the neutrons because those electrons are so teeny tiny. With nitrogen 15, though, this is our heavy nitrogen. It has an extra neutron, and so it's going to weigh more, but it's not going to be it's not going to be unstable, and so it's not going to give off this radioactive decay. And so it's still balanced enough in terms of protons and neutrons that it's not going to be radioactive, but it is going to be heavy, and this heaviness is going to allow us to help detect the DNA that is heavy. Another important thing about this labeling is that when you're doing this, you're basically making invisible changes in terms of the molecules around them. Because most of the activity is happening with those electrons, we're not touching the electrons, we're just touching the neutrons, and the neutrons are neutral and they're hidden in the central nucleus. Basically, the other molecules aren't even going to know anything's going on. So you can imagine if you stuck some big sort of bulky label or something on the nitrogen, then the molecules around it might treat it differently. But because this is kind of like a hidden labeling, you can label the DNA without actually changing how the DNA is naturally getting processed. So you can study these natural processes such as replication. And the way that they get this DNA heavy is by growing bacteria in media that contains heavy nitrogen. So basically the ammonium supply in the bacteria food is going to be having this heavy nitrogen. Because DNA contains nitrogen, so in these bases, they have these nucleotide bases, these have these nitrogens in them. And so if you grow the bacteria in this heavy media, then these nitrogens, the bacteria are going to incorporate this nitrogen into their DNA, and their DNA is therefore going to be heavy. If they then switch the bacteria to a normal media, so with normal nitrogen, this light nitrogen, then any new DNA that's made can only be light. And then they can look and see where the heavy DNA ends up. So just a technical note, so how they change the media, basically instead of dumping off the media and then adding new medium and potentially losing cells in the process, what they did was that they grew these bacteria in this heavy media for 14 generations. So they're swamping out any of the light nitrogen that these bacteria were originally grown in. Then to change the medium, they added a 10-fold excess of the light ammonium chloride along with the pre-made adenine and uracil, or the adenine, guanine, uracil, and cytosine in their second experiment. So basically, they're adding the raw source of ammonium in the light form, as well as the pre-made nucleotides containing the light nitrogen, and they're adding a ton of it. So it's going to swamp out the heavy stuff. And then each time after that, now they're adding more medium that has those the light nitrogen and these nucleotides. And so therefore they are able to switch the medium without actually having to try to remove medium and add new medium and potentially be losing cells in the process. This way they can make sure that they are not losing anything, but they are swapping out the media by swamping out the media. When they actually wanted to test the DNA, to get these DNA samples, they need to get the DNA out of the cells. So they lysed the cells, so they broke them open by adding sodium dodecyl sulfate, so SDS. This is the same thing that we add to our proteins when we're doing an SDS page to denature the proteins. SDS is a detergent, so basically it's going to kind of mimic the lipidy membrane surrounding cells, slip in there and disrupt things and break things open. It also has high salt and EDTA that's going to help destabilize the membrane. So this is all going to promote these cells breaking open, and then you could take a sample of it. So they need a way to be able to separate these DNA duplexes by their weight and be able to see how much of it is heavy and how much of it is light, and various things like that. And we'll go over this scheme in a minute. But we need to be able to do this at different generations. And so they're using bacteria, which is really great for this because bacteria, they have a fixed doubling time. So you can know because they're growing in this like exponential thing, you can kind of say, okay, at this point, it'll take them this long to go through one replication cycle. So if I take a sample at this point, then I know that it's been one cycle. And then if I take it at twice as long, then I know it's been 2 cycles, 3 times as long, then it's been 3 cycles. And so you're able to separate the DNA at these different generations. And then when you get the DNA, now you can separate those helixes and you can look and see if they're heavy or light or a mixture of the two. And so with these experiments, we're keeping these helixes together. So if you have a light strand, it'll light, you'll have light. If you have light heavy, you'll have light heavy. And if you have heavy, you'll have heavy. But you're not going to have these strands split apart. Later, we'll see something where they do denature the DNA, so they do pull strands apart, but they're not doing that here. Here, they're just separating them by their weight, or they're more technically by their density. And so they're doing this density gradient centrifugation. So how it works is that it's going to use the cesium chloride gradient. So cesium is heavy. So the further down in the periodic table you go, the heavier it is, the more protons it has. So cesium is this heavy element. And so if you have cesium chloride, so the chloride is this counter ion, so the cesium is going to hang out. It's going to basically give an electron to the chlorine. So you have cesium chloride, and this is a salt, so it's a neutral combo of a plus and a minus charge. But then when you dissolve it, these can come apart. But basically, you get the cesium is really heavy, and so it's going to start sinking when you put this in a centrifuge. So a centrifuge is centrifuge is just one of these machines that's going to spin really fast. The type of centrifuge that they're going to be used is going to be a spinning bucket centrifuge. And so it's something like this, where instead of a fixed angle where the tubes are held in one place, there's kind of these swinging buckets. And so this is going to have the consequence we'll see when we're spinning it, is that the force is going to be kind of sideways. So we're going to have to be thinking about things being horizontal instead of up and down. But the concept is the same, is that when you have a centrifugal force applied, it's going to the heavier things are going to be drawn towards the bottom or away from the force type of thing. So this is going to be, when we have our sideways tube, it's going to be like all the way at the end. But if we consider it like this for now, it's basically going to sink, but then you also have this idea, this diffusion going on. So diffusion is basically the random movement of particles, and on net, it leads to particles moving from a place where they're more concentrated to a place where they're less concentrated. But basically, it's just molecules moving around randomly and trying to spread out, because nature likes entropy, so like randomness or disorder. So these molecules are moving all around. Their movement is going to be inhibited by this centrifugal force. And so basically they're getting pulled down, but they're also trying to like diffuse away. And so you get this balance where you're going to get an equilibrium where things are going to end up at their like buoyant density, where the force that's trying to pull them down from the centrifugation is going to be counterbalanced by this force like diffusion. And so you're going to end up with these staying in a single spot. And so if you make these cesium chloride gradients, the same thing's going to happen with the DNA. The DNA is going to go to its spot where it's going to have that equilibrium reached in the cesium chloride gradient. And so the heavier it is, the more liquid nitrogen, the more heavy nitrogen it has, the further it's going to sink. And so therefore, if you're able to see where the different bands are, you can get information about how heavy and how light they were. And so this is what they did. So in the beginning of their paper, they do like a kind of proof of concept. So they show how they can use a cesium chloride gradient to concentrate DNA by density. So if you start with this technique, you make the gradient at the same time that you're making the DNA stack. So it's not like you're preparing the gradient and then you're sticking the DNA through it. Instead, it's this other type of like centrifugal density gradient centrifugation. So what they do, it's not called like isopenic. So basically what they do is they take the cesium chloride and they have the cesium chloride, it's all over the place, and the DNA is all over the place. And then what they're going to do is they're going to centrifuge it down, and the DNA is going to start moving. So you have these forces. You have the force where you have the centrifugation kind of pushing it this way, and then the diffusion pushing it this way. And these are going to balance out, and you're going to get the DNA to concentrate in a single band in this gradient. And so this is hours, so you can see it took them 43.5 hours. So it's not a fast process, but it gets concentrated in a band corresponding to its density. Then what they can do is they can use a densitometer to basically quantify the band darkness. So they can, the reason what we see darkness, the reason why we're able to see anything is because there's a UV detector. So DNA can absorb UV light. And so then they can kind of scan these and see where light is being absorbed, representing the location of the DNA in their gradient. And then they can use this densitometer to basically say, okay, how dark is this one? How dark is this one? And then they plot these out on these graphs. So the position in the gradient is going to be plotted as a peak on the graph. And the further to the right on these graphs, the heavier things are. So like the heavier, this is the heavy nitrogen, and this is the light nitrogen. This is going to get a little complicated when we talk about the denaturation, because instead of just like light heavy, it's actually like this density idea. But we're not there yet. So basically, think if you see a peak on the right, this is going to be the heavy nitrogen. If you see a peak on the left, this is going to be the light nitrogen. And this is what you see if you just have heavy or you just have light. But what happens if we actually do this with our DNA and replication and the different generations? What would we expect to see? So in these copying schemes, in our conservative, we would start with all heavy, heavy. We would then go to one heavy, one light, and then we would have that one heavy and then all the rest light. But in no case would we have any sort of mix between the two. With semi-conservative, this is here we would have, our original would be heavy, heavy. We'd split and we'd have two heavy lights. And then in our next generation, we'd still have the two heavy lights, but in this case, it would be a new second strand with the heavy. And then we'd have two lights. But in the distributive model, we would have since the very beginning, we would have a mix. And then over time, this mix, you would have less and less of the heavy because you're kind of diluting it out. But each of the pieces would still have a little bit of heavy. So if you had some sort of conservative model, you're always going to see these two distinct bands, one for the heavy and one for the light after you get your first generation. With semi-conservative, you're going to get that you would have, so in the beginning you would have heavy, heavy. The first generation you would have heavy, light, you would have two, you would have all just like a single band though for one heavy and one light strands, but these are staying together, so you get a band of intermediate weight. And then after that, you would have those bands with the intermediate, but you would also start having bands with light, light. You would continue to have this intermediate bands, but over time, there would be more and more of the light, light along with it. You can see that you would not be able to distinguish between semi-conservative and distributive at this first generation, because at this first generation, both of these would have in each strand, each duplex would have half heavy, half light, except in distributive, it would be kind of broken up between the strands, whereas semi-conservative, you would have one heavy strand and one light strand. And so in order to differentiate between these two models, they'll have to go to the second generation. So that was what we would expect, and what did they actually see? So here you can see that this is their generation 0, generation 1, 1.9, generation 3. And then at the bottom, they have their kind of markers. And so this is mixes where they have a known amount that they mix together to show you where you would expect to see different things. So if you had all light, you would have these bands, a single band here. If you all had all heavy, you would have a single band here. And if you had something where there was half light and half heavy, you would have a band in the middle. And so what would you, and this, I say they're controls, but they're actually coming from their different generations, but mixes of them to show you where the different rates run. So here in generation 0, you can see that we have a band over here to the right, and this is representing an all heavy band, which makes sense because we started with it grown in all heavy media. Now at generation 1, you're getting that you have all in this kind of middle spot because you have one heavy and one light strands. As you go up in time, you're keeping that middle strand, but now you're also gaining a light. And so the middle strand is going to start to kind of fade, and the other one's going to get darker because it's getting kind of diluted out. And so if you take like a constant amount of DNA or something, then you're going to end up with more of the light in proportion to the heavy light, but you're still going to have that heavy light persist throughout because that original copy is still going to be there, even though you're having a lot more of the new copies be made. And so this is what they saw, and then this is how they were able to interpret their findings. And then based on their data, they proposed this model supporting the semi-conservative manner of DNA replication, where the original parent molecule gets split, each is used to make a template of the other, and then those get split and each is used to make a template of the other. This theory, if this was true, which we now know that it is, it would require that the strands of the DNA helix be pulled apart. And this is, some people thought that this would be very, very unlikely because these DNA strands are held together by like these hydrogen bonding, which is how we can get this base to base complementarity. So you have these attractions holding the DNA strands together, and it seems like it would be very energetically unfavorable to actually pull them apart. And so to help show that these strands actually do get pulled apart, they did some thermal denaturation studies. In addition to helping show that these strands get pulled apart, it helps show that the strands really are in that semi-conservative model where one strand is all heavy and one strand is all light, as opposed to some sort of hybrid from a distributive mix. So one thing to note is that the sharpness of the peak is going to tell you information about the molecular weight. So if you have a sharper peak, that's going to be like a higher molecular weight. And if you have a broader peak, that's going to be a lower molecular weight. So if you have double-stranded DNA, you would expect it to have a higher molecular weight. It would have like twice the molecular weight of the single-stranded. And so you would see a sharper peak. And then if you have it be the strands pulled apart, so you have single-stranded DNA, then you would see a broader peak. And in fact, this is what you would see. So this is showing that what they did here was they took this bacterial lysate, so they split open bacteria that had either uniformly labeled N15, so heavy nitrogen or light nitrogen, and then they heated it at 100 degrees Celsius for 30 minutes. And then they see that the apparent molecular weight of the heated bacterial DNA is reduced to approximately half that of the unheated material. And so this is consistent with the strands coming apart and there being like single-stranded DNA. So in addition to the band broadening, you see a shift when you denature the DNA to a higher buoyant density. And so the single-stranded DNA has a higher buoyant density than the double-stranded DNA. And the reasons are kind of complicated, and huge thanks to Dr. David States, who answered my Twitter question about it and helped explain it to me, and also directed me to this paper. But basically what happens is it has to do with the cesium counterions. So DNA has that phosphate backbone. If you have a double-stranded DNA, it's more likely to be protonated than in single-stranded DNA. Basically, the pKa of those phosphate groups is a little different in the double-stranded context. So in the single-stranded DNA, you have more of the cesium counterions counterbalancing the negative charge of the phosphate, as opposed to the protons. So if there's a proton there, that's really small. If there's a cesium there, that's going to be really big and heavy. And so when you have the single-stranded DNA, you're going to have that backbone kind of coated in cesium. And this is going to add to the density of this complex, because what this technique is doing is it's measuring the density, the buoyant density of like the DNA like molecules surround with its like ions that it's bound to, that aren't like just like free to move around. And so in your double-stranded DNA, you're not having as many of those cesium counter ions because you're having more of the protons, whereas in with the single-stranded DNA, now it's going to be less protonated, more cesium, and this is going to make it heavier. Basically, you're able to see what you would expect with the double-stranded DNA, with the heat denatured DNA. And then what you could do is they take a hybrid DNA. So they take this heated lysate where they have bacteria grown for one generation and then light media. So this is like the generation one. And so you would expect that there would be this mix of heavy and light strands. And when they denature it, they see that they get a peak corresponding to the locations that they would expect if they had heat denatured light and heat denatured heavy. And so this is basically showing that when they take this hybrid DNA and they heat it up, the strands are splitting apart, and the splitting apart to give you one strand of all heavy and one strand of all light. And so this is a really important set of experiments, and they're really fundamental in how the idea of DNA replication is understood and how we're able to have each copy of DNA, each like cell pass on its full copy of DNA in the semi-conservative way. There's also some companion articles with more historical background if you want to know more about that. So hopefully that helped you understand these classic experiments and recognize how cool biochemistry can be.